First, I'd like to thank David and Ran for the invitation and thank for David's very nice introduction. It's very an honor to give a talk here, and I'm very happy to have this chance to introduce our recent work on timescale localization and signal propagation in the large-scale cortical network. So this is a joint work with Songting Li. Now he is a postdoc at HHMI, and also my student Zhenyuan Jing at SUTU, and also Xiaojing Wang at NYU. So I'd like to start my talk by introducing the phenomena of timescale hierarchy. And we are all very familiar with the concept of spatial receptive fields of visual neurons. That's a specific region where this neuron will respond to. And experiments have shown that the size of the receptive field actually will increase along the visual hierarchy. For example, this LGN neuron's receptive field, the circle is usually smaller than the V1 neuron's receptive field, which is the elongated bar. And we can expect that the V2 neuron's receptive field is even larger than V1 neurons. And the mechanism of this increased receptive fields along the visual hierarchy has been well understood many years ago by Huber and Wiesel. It's due to the convergent connectivity. For example, we have many LGN neurons connected to one downstream V1 neuron in a convergent way, so that V1 neurons receptive field will be the summation of the receptive field of this LGN neurons, so it's bigger. So this is considered as the spatial property of a V1 neuron's response. And the question is, what about the temporal property? Do we still have similar phenomena or not? So this question is recently under active investigation. For example, this is an experiment done in the Allen Institute. They use this neural pixel to record neural activities in multiple regions of the mice cortex, while the mouse is watching some visual stimuli like drifting grading. And here's the raster plots of neural activity. The x-axis is time and y-axis is the neural index. And we can see that for these 8 areas, this neural activity show different patterns. And if we want to study the temporal response of these visual neurons, and we can compute the autocoloration of the neural activity in each area. And then we do average. So here, each different color corresponding to different area. And it's the population average autocorrelation function. And we can see they actually have a very distinct decay rate. And if we fit this autocorrelation function, we can, using an exponential function, we can define the response timescale of each area's activity by using this decay rate tau. And for each area, we can also assign a hierarchical number to it according to the position in the visual pathway. For example, V1 has the lowest hierarchy, and sorry, here LGM may be lower, and it will increase along the visual pathway. And then we can see that this time scale will actually increase along this hierarchy. So that means if an area sits in the high order position, it's a high order area. It usually has a slower time scale. And this phenomena is called timescale hierarchy. The time scale basically will increase along the cortical hierarchy, just like the spatial subfields. And it's very general, not only being discovered in mouse, but also in macaque and in human. And it is considered as a very prominent feature, an important feature of the cortex, because we can think about in sensory areas, when these areas receive the external stimulus, it will respond to this stimulus as fast as possible and then transmit this process signal to downstream areas for further processing. But for high order areas, we actually require these areas to slow down a little bit, because this area usually is for high order cognitive functions like decision making. So it requires a certain time window to accumulate evidence and to integrate information and then make a decision. So it's very important for them to slow down. but the mechanism is not very clear yet. So we'd like to study what's the mechanism underlying the phenomenon of timescale hierarchy by developing a multi-regional model of the macaque cortex. And this model was initially developed in Xiaojing Wang's lab. It includes 29 areas, including low-order sensory areas and also high-order association areas. And it has two features. One is that this model is based on the latest macaque connectome derived from Harry Kennedy's lab. And the other one is it considers area heterogeneity, which means that each area's dynamics is different. And later I will explain in the few slides how we model this part. Okay. So a classical way to model each area's activity is to use the mean field approach, like this Wilson-Cohen type model. We just view all the excitatory neurons into an E population and all the inhibitory neurons into an I population, and they interact with each other. And with no interaction, then for this E population's fine rate, it will decay to 0. And by including the interaction, we have the self-excitation and the inhibition from I population and external currents. And this current will go through a transfer function to convert to this neuron fine rate. Here, we just use rectified linear as a very simple approximation. And this is slope here. And similar thing for this I population. But we know that area is different in many aspects. For example, for this visual system, this diagram drawn by Van Essen and Van Essen, and we know that this visual input will go through these areas one by one in a hierarchical way. So recent experiments have carefully examined which area is higher order based on two anatomical features. One is that if the connectivity is from a low order area to a high order area in a feed forward way, then this connection usually comes from the superficial layer of the upstream area. But if it's a feedback connection from a high order area to a low order area, it usually comes from the deep layer of this upstream area. And based on these two facts, we can derive a value. This we call the hierarchy score, this value for each area. So if this area is has a very small hierarchy value, then it's a low order area. And we can see that for this early visual areas, there's a clear hierarchy trend, and then it flattens out afterwards. So that's how we quantify each area's hierarchy. And how do we model this hierarchy in the dynamical equation? So there's another experiment done in Elston's lab. So they basically measure the dendritic spine numbers for a pyramidal neuron in different cortical areas. And they have found that for early sensory areas, the spine number is very smaller than those in this high order areas like prefrontal cortex. And there's a clear positive correlation between the hierarchical position and the spine count for all these measured areas. And we know that spine is the place where this neuron receives excitatory inputs. So larger spine number indicates that this neuron receives more excitatory inputs, so the excitatory connection will be stronger. So this allows us to scale all these excitatory connectivity strengths by this hierarchy value we have derived in these anatomy experiments. And because this hierarchy value hi ranges from 0 to 1, so in order to avoid vanishing connection strengths, we take the form like 1 plus eta hi here. Okay. So now we have the model with inter-area heterogeneity. So basically, it's a Wilson-Cohen type model. And for this old excitatory coupling strengths, including this self-excitation within one area, and also this long-range interaction from another area to a target area, we just scale with this hierarchy score. And this is a very simple model, but is it enough or sufficient to capture the phenomena we're interested in? Then we can do the simulation. We just stimulate V1 with a pulse input. And then we can see this V1 will increase its response and when the input is on in a very fast way, and then it decays to its resting state very fast as well when the input is off. But as the area's hierarchy goes up, we can see for this top hierarchy area, like 24C, it increases its response very slow and then decay also in a very slow fashion. So indeed, we can see a hierarchy of decay time scale here in this simple model. And if we give a noise input, like white noise drive to V1, we can see V1's fluctuation is very fast. But for high order area like this 946D, it shows a slow activity. And if we do this autocorrelation as we have done in the experiments, we can see the decay rate is very different across areas. And if we arrange these areas in a hierarchical way, we can see roughly an increase of time scales here. But it's not the perfect, it's not strict, but there's a clear trend here. And since this model can generate this phenomenon we're interested in, a natural question will become, what's the mechanism? Can we analyze this model to understand the emergence of timescale hierarchy? So the phenomenon is not trivial in the sense that this network connectivity is very dense. So you can imagine if V1 has a very fast time scale, but this high order area has a slow time scale, there's a large chance they will be connected to each other. So the time scale will be easily mixed. But why in such a dense network we still see the time scale hierarchy is a non-trivial question. So we'd like to perform some analysis into this model. And we first rewrite this model into the vector form. And we just write it denote nu as the final rate vector for all these areas. And W is the connectivity matrix. And we have also the external inputs. And W has two parts. One is the inter-areal connectivity that only describes the connectivity within each area. And also the other part is the inter-area connectivity. That's the long-range interaction. And all the model parameters are just based on some previous studies and the FLN, this so-called fraction of labeled neurons. That's the one way to measure the long-range connectivity. I didn't mention the detail here, but if you're interested, I will explain afterwards. But this you can just think of it as the number of neurons projected from one area to another. So it's scalable to this connectivity stress. And with such a linear model, we know mathematically we can solve it analytically. So the response of this, for each area, the vector, it should be the weighted sum of all these eigenvectors. And the weights are time dependent. And it's determined by the corresponding eigenvalue. So that's the simple linear algebra for differential equations. And from this expression, we can gain some intuition about why this model can have timescale hierarchy. Think about a toy model with only 3 areas. And these are 3 eigenmodes, and the corresponding coefficients are time dependent, are determined by the corresponding eigenvalues. So V1 has a coefficient that fluctuates very fast, and V3 has a very slow coefficients. And the response would be the linear summation of these 3, but weighted with the coefficients. Okay, so if you want this node, this area to maintain a very fast time scale, you will require the eigenmode corresponding to the fast time scale will have a very large elements here. So the multiplication will have a large effect to have this faster fluctuation. But for the other eigenmodes, you require, for example, these elements to be very small, such that this coefficient to multiply these elements will have little effect to affect this activity here. So similarly, for this area, if you want this area to have a very slow time scale, you will require these elements to be large so that this coefficient, slow coefficient, will come into the activity. But for this component and this component, you want this to be small. Okay, so it will not be affected by the fast coefficients here. So in short, timescale localization will require these eigenmodes to have very localized property, meaning that only a few elements will be non-zero, significantly non-zero. Okay, and also the non-zero elements should be non-overlapping. So that's called basically orthogonal to each other. So that's the picture or the intuition we have gained from this analytical solution of this linear system. Okay. And then we go back to check our model by computing its eigenmode. And here's the eigenmatrix. So each column is the eigenvector. We do see that for each column, only a few elements are non-zero. So it's very localized. And these non-zero elements are not overlapping. So it's basically like a diagonal matrix with some perturbation. Okay. So we do observe this localization and orthogonality of these eigenvectors for this excitatory population part. Okay. And then we have a puzzle because the network is very dense. So if we just randomly generate a dense network with a connectivity or sparsity like 65%, then the eigenvector will be very dense as well. Okay. So why in the dense network connectivity matrix we still have this very localized eigenmodes is a puzzle. So we start to think about the analysis. We first mathematically, so we first diagonalize the intra-areal connectivity by finding a matrix P. So we just do similarity transform to diagonalize this intra-areal part. So it will become a diagonal matrix. And the long range part will become some other matrix. Okay, we denote it as a sigma. That's a long range one. And so the picture is like, because there's an EI interaction, and if we interested in E, so we needed to get rid of this I effect. So we just do the similarity transform to disconnect this EI, but we needed to transfer the coordinates from RERI to UV. Okay, but the cost is we will have another additional interaction here. Okay. Oh, by the way, I forgot to mention. So in the original connectivity motif, the long range is only from the upstream's E to downstream's E and I. There's no long range I interaction. Okay, so that's also according to experiment finding. But now, because of this linear transformation, we will have additional two connections. And then we can find for the model parameters we use, there are two small parameters. One is called epsilon. This epsilon describes the distinct electrophysiological property of ENI populations. For example, they have different slope of Fi curve and different membrane time constant, so on and so forth. And another small parameter is called delta. And from this expression, we can see it depends on the connectivity strength marked by this red color. So there are two pathways for an upstream area to influence downstream area's E population. One is the direct excitation from upstream's E to excite downstream's E. And the other is the upstream's E first excite downstream's I, and then I inhibit the downstream's E. So that's an indirect inhibition. And this delta actually describes the detailed balance of these two inputs. For example, when this V4 receives input from V1, we can measure in our model the direct excitatory current, the red color, and also the indirect inhibitory current from I to E, that's the blue color. So they nearly balance with each other, leading to a very small net current here. And this is called detailed balance because it's not the balance of the total E input received by V4. Balanced with the total I input received by V4. But it's the pathway by pathway proposed by Vogels and Abbott many years ago, and they believe this state will be beneficial for signal gating. And here we also see this kind of regime. And with these two small parameters identified previously, for this new coordinates u and v, we can solve its eigenfunction. And we can actually denote, just write down all the terms, the diagonal matrix plus some long-range perturbation, and all these terms, the order. And we find that there's one term with the smallest order. So this term in the long-range connectivity matrix. And we can just be brave to ignore it, to set it as 0, because it's the it's the highest order. So we can set it as 0. Okay. And by doing that, we can decouple u and v now. Okay, so once we set that term to 0, the u equation does not depend on v anymore. And the u equation, we can see it's a diagonal matrix plus some long-range perturbation. Okay, that and because of these orders, we can see indeed this long-range interaction is a perturbation. It's one order smaller than this diagonal matrix. So naturally, we will have the u solution as the natural basis. This ek is like 1, 0, 0, 0 or 0, 1, 0, 0. This natural basis plus some perturbation due to this long-range interaction. Okay, and this localized eigenvector here plus some small perturbation actually explains well the localized eigenmode as we have shown in our simulation. But this so this expression holds with certain conditions. We can further solve the first order perturbation as the ratio between this long-range interaction and the spectral gap, the difference between two eigenmodes. And in order to make this perturbation valid, we will require this u1 to be at most order 1, right? So this can be viewed as a small perturbation. So in that case, we require this spectral gap to be larger than the perturbation element in order to make this u1 to be at most order 1. So we will require the big spectral gap. So difference should be big. And the difference is due to the strong area heterogeneity we previously set. Different hi will give different lambda here for each area. And the small perturbation in the numerator requires the detailed balance of E and I inputs. So you can think about if each area, the effective interaction between two areas due to this detailed balance of E and I is small, then each area will maintain its its intrinsic time scale, and plus this heterogeneity will help against this long-range connection. And if we numerically check the perturbation of this eigenmode expression, it's actually very similar as the numerically solved eigenmode pattern. So that verifies in this parameter regime, our analysis is valid. But later we feel that this explanation may require more refined characterization. It's not sufficient. It's not enough, because let's think about an extreme case. If the perturbation is very small, like it's perfect decoupled, then each area can maintain its time scale for sure. But because the long-range interaction is very weak or vanishing, then we will have a problem of signal propagation. So it's very hard for one area to propagate signal to the downstream areas. And then we have checked this case. For example, for some parameters that satisfy the previous condition we have derived, if we give input to V1, a pulse input, for downstream areas, we will see signal attenuates quickly, substantially by 5 orders of magnitude when it arrives at the high order area like 24C. So this is not reasonable in the brain. And so it seems that these two conditions are somehow contradicting to each other of timescale hierarchy and signal propagation. So in order to achieve reliable signal propagation, one solution is to tune the parameter to another regime called global balanced amplification. Basically, it's just to increase the E to E connection between two areas. Meanwhile, we increase the I to E connection within one area. So the increase of strength is in a balanced, EI balanced way, but the effective interaction will be increased. So in that case, we can see when we give a pulse input to V1, indeed, in high order areas, the final rate can be a few hertz, so which is reasonable in the brain. But in that case, because the effective interaction between two areas is increased, so time scale will be mixed in this case. We can see if we plot the time scale across areas, there are only two time scale pool. One is very slow and one is very fast. So that's not consistent with the experimental finding. So it seems that, as I mentioned previously, so these two properties are contradicting to each other in some sense. If we have very strong connection between two areas, we will allow reliable signal propagation, but timescale will be mixed. But if we have weak interaction between two areas, timescale can be maintained for different area using its intrinsic timescale, but signal propagation will be a problem. So how to achieve both? We try to study this question with starting with a toy model. So in this toy model, we only have two areas or two nodes. One upstream area just send input to a downstream area directly with the coupling strength mu EE. And we also use this linear first order dynamics to describe its interaction. And here we set the downstream area to have a very fast time scale, and the upstream area has a slow time scale. And when the coupling strength is very small, we can see the downstream area's response is small, but it can maintain its faster time scale. It reaches steady state quickly. And if we increase the coupling strength, in that case, we will increase the downstream area's response, but the time scale will be interfered by the upstream's time scale. We can see both the upstream and downstream area seem to have the same time scale now. Okay, so if we quantify the interference of time scale from upstream area, we can fit the downstream area's decay response using two exponential functions. One exponential is with the downstream's intrinsic time scale, and the other exponential is with the upstream's intrinsic time scale. And then we just compare the coefficients in front of these two exponential functions. So if the upstream area have no effect on the downstream area, then this coefficient will become zero. The interference index will become zero. But if the downstream area's time scale is dominated by the upstream area's time scale, then we will have very large coefficient here compared with this one. So interference index will become 1, or at least approaches 1. So now we increase the coupling strength between the upstream and downstream areas. Then we can see the downstream area's peak response will increase. But meanwhile, the interference index will also approach 1, which means for very large connectivity strengths, the upstream area's time scale will dominate downstream's area's response. So in this toy model, it's not possible to achieve both. So we also realize that in the brain, we also have I neuron, right? We have inhibitory neurons. So let's add I inhibitory neurons to the model by adding another equation here. It's also a linear equation. And here, for simplicity, we only consider the interaction from I to E. It's not realistic in the brain, but it's enough to illustrate the idea. So let's start with this toy model. And then with certain parameter regime, if we increase this mu EE, the E to E coupling, we can see the downstream area's response will increase. Meanwhile, it can maintain its very fast timescale, not be interfered by the upstream area's slow timescale. And if we compute the interference index in this case, when we increase the interaction between E and E, the maximal downstream area's response will increase, but the interference index will maintain in a very low level, close to 0, indicating perfect, almost a perfect timescale localization. And if we give some sinusoidal input to the upstream area, and because the upstream area has a very slow timescale we set previously, so it will low pass filter this external oscillatory input, so with some delayed phase. But for the downstream area, we can see the response, the delay actually becomes less. It can faster check the external inputs, although the external inputs is only given at the upstream area. But in this case, it seems that the downstream area directly receives this external inputs, so it can faster check it. So why is that? So next, we can perform some analysis to understand this seemingly very simple linear system. Now, we first do some non-dimensionalization for the variable of final rate. And then we can derive an effective post-area dynamics for this downstream E population. We can take the derivative of this E dynamics and then using the second I dynamics to eliminate the variable I here. Okay, so it becomes a second order dynamics for the downstream E population. And here we have a small variable again showing up, epsilon here, which depends on the ratio between the E and I time constants. So it also reflects the difference of E and I population in this case. And this is a small variable here. And that will make this problem become a classical initial layer problem in the perturbation theory, meaning that within a very short time window at the very initial point, there will be a sudden change of this neural activity with a distinct time scale. But that region in many problems is very important, but here in our case, it's not very interesting. So we just directly set epsilon into 0 to further get rid of this high order derivative to become the first order dynamics to approximate this E dynamics here. And we can see that approximation is actually very accurate when the upstream area receives some external noise drive. So the area is very small. And from this very simple effective post-aerial dynamics, it's interesting to see that the drive to the downstream E population has two parts. This is the effective drive now. So one is depends on the activity of the pre or the upstream area directly. And the other is the change of activity of the pre area. And both play a very important role. So let's first look at the first term. So let's consider when the upstream area receives some stationary input, it reaches a steady state. So the time derivative can be ignored first. And then the downstream's inputs will be scaled by the coefficient here in front of the upstream area's response, right? So if the coefficient is larger, which means the downstream area's input is larger, so the response will also be larger. So this coefficient can measure the signal propagation of this very simple motif. With larger MSP, the coefficient, we call it signal propagation measure. With larger this coefficient MSP, we will have larger response in the downstream area in the steady state case. Okay. And indeed, we can see this in our numerical simulation. By increasing MSP, we will have the larger this maximal activity of downstream areas, E population. And if the upstream area receives some temporal fluctuation part inputs as well, then we can use the third equation, the upstream's its own dynamics, to plug in here. And then here we have REJ, the upstream area's response as well. So we can combine these two terms. We will have a new form of downstream areas response. So it is influenced by upstream areas response that could be time dependent and if the external input received by the upstream area. Okay, so now we can see if this coefficient becomes 0, which means the downstream areas will directly receive the external input to the upstream area, although the input is given in the upstream area, right? So that means the input to the downstream area will have no information, no temporal information of the upstream area's dynamics. So the time scale of upstream area will not enter or interfere the time scale of downstream area. So this coefficient is very important. We call it time scale localization measure. If it's 0 or close to 0, the upstream's slow dynamics will not enter the dynamics of the downstream area. So indeed, we can also see this in our simulation with small MTL, the interfering index is very small for the downstream area. And for the large scale network case with multiple regions, we can generalize the metric for MSP and MTL. Basically, we just compute all these measures for all pairs of this area, and then we just do average. Okay, so this averaged metric for MSP and MTL just quantify the average ability of timescale hierarchy and timescale localization, and this average ability of signal propagation. And so we also use a measure called IPR, this inverse participation ratio, to quantify the degree of timescale localization in this network. And this this measure actually was initially used in quantum physics, but the idea is very simple. We just calculate the force power of the elements in each eigenvalue, in each eigenvector. So it also quantifies the how localized each eigenvector is, and then we just do average for all eigenvectors. And then we can draw a phase diagram. We scan the parameters of the model, and for each parameter, we can compute one MSP value and one MTL value. Meanwhile, we can compute its eigenmode and to compute IPR to see the eigen timescale localization ability. And we can see that timescale localization indeed is mostly dependent on MTL, but not MSP, because once MTL is fixed, if you look horizontally, this IPR is a constant value almost. And to quantify signal propagation, we just give input to V1 and then measure the response in 24C, which sits in the highest highest position in the hierarchical axis. And then we can see for signal propagation, it mainly depends on MSP rather than MTL, because if you look vertically, once you fix MSP, the signal propagation, the response of 24C is almost a constant. And with these two phase diagrams, we can mark the dynamical regime we have discussed previously. The first one is the weak GBA. We have very good timescale localization here, but with the signal propagation, we have some problem due to this very weak effective connectivity. And we also have the strong GBA. We have very good timescale signal propagation, but we lose timescale localization here. And in the phase diagram, we also identify a new regime. In this regime, we can see that both timescale localization and signal propagation can be achieved. And if we give a pulse input to V1, for example, we do see each area will respond to this pulse with its distinct time scale. So the time scale, if we arrange it along this hierarchical axis, will show a clear trend of gradients. And the signal attenuation is also reasonable, about 2 orders of magnitude, compared with the previous case, about 5 orders of magnitude. But can we understand more about the mechanism? What's the physical intuition about this regime we have previously identified? So actually, we can understand the picture using, go back to this very simple motif. We have one upstream E project to downstreams E and I. And if the upstream response, if we decompose this upstream's response into its mean part and the time fluctuation parts, and because everything is linear, right? So it's a linear model. We can compute the total current received by the downstream E population. So it also has its mean part and the temporal fluctuation parts. And in the mean parts, the previously measured MSP appears here. And in the fluctuation part, we have this MTL measure appears here. So the mean total current actually is contributed by the summation of the direct excitation currents and the indirect pathway inhibition currents. So if we have a large MSP, which means the mean is large, so the two currents, E and I, will not cancel in their means. So leading to a large mean, so the signal can propagate. But MTL, when it's small, indicating that the temporal fluctuation part will cancel with each other. Then the net current will have a time constant almost independent of upstream area's time constant. And this can be shown by computing the autocorrelation of these currents. So if we compute the autocorrelation for the E current and I current separately, we can see that it has a very slow decay, right? So indicating the upstream's areas E and I inputs, it has a very large time constant. But for the net current, if we measure it from simulation and then compute its autocorrelation function, we can see it decays very fast. So it's not contaminated by the upstream's slow time constant. And if we compare with the control case, the dashed line, that's the okay. So here's not case. Okay. Anyway, so from this toy model, we can understand the picture. And now go back to the full model. Some people may argue that it's a linear system, right? But the brain may not be linear. It's just a toy model. So the motivation of our study with the linear system is that for linear system, the time constant is well defined. It's the negative inverse of the eigenvalue, right? For nonlinear system, the time scale could be state dependent. You need to linearize it near some point. But we can also generalize our results to a nonlinear system with some spiking neural model. So for each area, now we include 1,000 neurons with the EI ratio like 4 and 1, 4 to 1. So each area follows a integrated fire type model. And for this model, if we average the neural activity, we can see also a clear hierarchical time scale and also a reliable signal propagation as well. And interestingly, if we compare our model, this spiking model with this linear model, there is very high correlation, either both in this final rate and also the time scale. So indicating that in the mean field sense, or the coarse grain sense, the linear model is actually a very good approximation, at least in this regime of this spiking neural model. And then we selected a few areas to see that indeed, when V1 receives some white noise drive, they show different time scales. And if you compute this autocorrelation function, we can see that either E and I inputs have very slow time scale from upstream area, but the net input has a very fast time scale because the temporal part cancels with each other, but the mean component is not canceled, so that signal can still propagate along this pathway. And we also looked for this experimental evidence about this IFP regime we identified. Unfortunately, at this stage, we don't have this macaque data activity recording, but we do find some open public Yeah, it's the last slide actually. So we just determine the parameter constrained by the neural activity recorded in the experiment, and then with the determined activity parameter, we can simulate this model. We can see each area will have different time scale, and the time scale, simulated time scale, also well correlates with agrees with measured experiment. And similarly, in the experiment, they have used this optogenetic stimuli for each area, and then measure the response for different areas. And we can do the same in our simulation with a determined parameter, and we can well predict the neuronal response for different areas. And then with the determined parameter, rather than fine-tuned, so we can plot the phase diagram, and we can see for this IPR timescale phase and also the signal propagation phase, we can mark our parameters or model here in the screen dots here, which also shows this IPR as we have previously discovered in the macaque model. So from the marmoset model, it seems we have some indirect support for this IPR regime. Okay, so in summary, we have identified a novel regime termed as interference-free propagation, which allows the coexistence of timescale hierarchy and signal propagation. And the IFP regime exists also in the spiking neural model, and also is supported by the marmoset experiments. And these two are the references of today's talk. And at the end, I'd like to thank David again for the invitation. I can only do this virtual handshaking. I cannot find the recent one, but this is also a nice memory. Yeah, and thank you all for your attention. Thanks.